All right. I think we will go ahead and get started. I'm just going to start off with a brief introduction. So, welcome to the Peptide Drug Hunting Consortium and XtalPi webinar. Um, we are in a Zoom webinar, so attendees do not have access to your microphones or your video. For question and answers, there should be a Q&A button at the bottom of your screen. Please use that instead of the chat to ask questions during the webinar. Um, you should be able to click the Q&A button, type in your question. Charlie Johannes, our president, will moderate the Q&A, and we will hold the Q&A after each presentation. So I'd like to get started and introduce our first speaker. If you want to share your screen. I'd like to present Dr. Xiyun Ye from the University of Tokyo. She's currently a postdoc fellow under Dr. Shuga. And yep,
Yep,
there you go. All right, thank you.
sounds good. Okay. Oh, then I'll start it.
Yes.
Okay. Yeah. Thank you, Daniel, for your kind introductions. So hi everyone. This is Xiyun. So I'm currently working in Suga Lab at University of Tokyo as a JSPS postdoc fellow. So today I'm going to share about our recent progress about the ribosomal elongation of a unnatural amino acid building block named trans-4-amino crotonic acid. And then we put this amino acid into a nascent peptide chain. And so this is not a so-called like terminal amino acid, but we actually put this amino acid as part of a longer peptide. So this would be the project I'm going to introduce for the later sessions. But just sort of because my PhD work is also related to peptides, so I'm also gonna briefly walk through some prior work that I did before I joined U Tokyo as a postdoc. So I obtained my PhD at MIT Panaludi Lab, and then throughout my PhD work. I work pretty closely with Calico Life Science. So it has been a great industrial collaboration where we would be able to sort of advance a technique called affinity selection mass spec. So basically, we run affinity selections using peptide libraries and then trying to find high affinity binders against protein target of interest. And then we're sort of using a size exclusion chromatography to separate bind versus non-bind binders, and then we use mass spec to decode these bind peptide fragments. And other than ASMS, which we design a lot of focus library trying to find matured peptide binders that with high affinity, I also work on some covalent warhead that we use selective covalent warhead, and then we sort of make covalent peptide inhibitors that can selectively and also specifically cross-link onto a target protein of interest. Beyond that, we also utilize the high affinity properties of peptides to generate selective lifestyle labeling reagents. For example, we trying to generate branched peptides that sort of have a more complex architecture of peptides, and then use this as a way to authentically mature the peptide, and then eventually we were able to label lifestyle just in 5 minutes. My interest of peptide actually began in my undergrad, where I started to work on total synthesis of daptomycin, which is a natural product in antimicrobial peptides. And then throughout my PhD, I work a lot with peptides, but that made me wonder what I can do further on. And I realized that the novel peptide discovery is also a very powerful tool that you could find a high affinity hit, a very important starting point in terms of the peptide drug discovery, where you could start with a high affinity de novo binders. And then you could sort of skip some of the affinity maturations with naturally existing peptides. So just to sum up like my background a bit, so I did MR display, and then during my PhD, I work a lot with ASMS, and then now I'm focusing on trying to generate macrocyclic peptide libraries by using MR display. But I also work on covalent peptide inhibitors and some high throughput method of generating peptides using flow chemistry developed in Penaludi lab before I joined U Tokyo. But I guess to this audience, I don't have to, talk a lot about why peptides such advanced modalities in terms of manipulating protein-protein interactions. But just, still a little bit recap. Peptides naturally they exist as PPI modulators. Um, and then peptides can bind to some challenging PPI interfaces that small molecular may be a little bit, weak towards these PPI interfaces. And peptides are also highly tunable. They can be made with easy, like you just need to make it on an fully automated peptide synthesizers, and then within, just 1 to 2 days, you can get a peptides. Also, peptides come with all these non-proteogenic or unnatural amino acid building blocks that are available and commercially, accessible. And then these amino acids are necessary to advancing the drug-like properties of peptides. In order to improve the stability profile, potency, level velocity, renal clearance probabilities, and also the rigidity solubilities, like properties of peptide central. So, so one of the angle that these our MR display people are thinking about is to bring this unnatural amino acid building blocks as part of the de novo discovery campaign, so that we could incorporate this unnatural amino acid when we um, like during the de novo discovery, so that we can sort of like pre-install unnatural amino acid on a peptides. So the inspiration of our recent JAX publication is to translate some of the naturally existing gamma amino acid and in trying to incorporate them in the ribosome synthesis method. So, for example, we've been observed these gamma unsaturated amino acid in some of the natural products. Just to name a few of these natural products, you can tell by their structures. These gamma unsaturated amino acids are sort of like actually they present in nature, and then they present in different format of the gamma amino acids. For some of them, they bear a side chain that looks like, for example, alanines, and some of them are having some even weirdly looking like structure that having double bonds, and some of them have a valine looks like gamma amino acid. Some of them also look like tyrosine. So the key challenges that we've been observed with these non-natural amino acids is that in nature, these amino acid has only been produced as part of the non-ribosomal produced peptides. So those peptides have been made by some gene clusters. So basically, these about these gene clusters or enzymes have a low substrate tolerance, which could limit the structure diversity when you're trying to produce a highly diverse peptide libraries in order to generate more drug-like libraries. So it is actually very hard to reprogram them. So if you read some of the natural product paper, you'll realize that the number of peptide library they can produce is somewhat like limited beneath 100, like 100 moleculars in each discovery campaign. So what I thought about this is that why don't we just extract these building blocks from nature, and we learn from nature, and then we make these building blocks, and we're trying to incorporate it as a natural amino acid building blocks in our margin supply. And our ultimate goal is to incorporate this amino acid as part of the peptide elongation, so that we could generate macrocyclic peptide libraries that bearing all these gamma unsaturated amino acids. So like our ultimate goal is to build these gamma-saturated amino acid contain peptide libraries that in total have more than 1 billion members. So the challenges here about gamma amino acids is that we can use flexism. Like flexism is our, the ribosome that can catalyze easily the amino acyl tRNA reactions that ultimately we would generated amino acyl tRNA that basically you can incorporate a wide range of different unnatural amino acid building blocks. But the problem that we observe is that when we, even though we had, this highly accessible AATRNA, but we still observe ribosomal stalling when we're trying to synthesize peptides using the ribosome, in vitro translation systems that using the ribosome to produce peptides in vitro. We kind of constantly see a drop of truncation. Basically, the peptide growth would stop just before the unnatural amino acid building blocks. And then we will see a lot of fragments. So you would no longer see the elongation of the peptides. And we also observe a low translation efficiency because the ribosome was not designed to incorporate the gamma amino acid. So that's why we need to customize the system in order to force the reaction to proceed further. And just a slight flavor here, the reason why I chose the unnatural gamma amino acid is because the gamma amino acid would constantly form, like it would hydrolyze when the double bond is not present. So the double bond here actually give the structure a little bit of rigidity. So that would present the hydrolysis of the AATRNA. So that's also one of the reasons why I chose to work with gamma-unsaturated amino acids. So the aim of the study is trying to fine-tuning the translation system, make high efficient translation, and then also trying to build up the codon toolbox so that other groups can sort of fit in this unnatural amino acid into different codons. And then we're also trying to test the limit, like the upper limit of the ribosome synthesis that we would love to see, like how many gamma amino acid that we could put into the elongation reactions. So initial task with this translation, it's kind of it's like we come with surprise, but they also come with some drawbacks. But you can tell that you will constantly see the N-terminal drop-off fragments that you won't see the elongation of the gamma amino acid and also the elongation beyond the gamma amino acids. So you constantly see the N-terminal fragments. So we thought about how we can improve that. So in Suga Lab, our associate professor Kato-san, he sort of developed a artificial tRNA, and this is inspired by some of the I think E. coli system where they have a tRNA called Pro1E2, and this tRNA can have the better, so it can actually form tighter binding towards some of the elongation factors, namely EFP and the EFTU. So these elongation factors would bind to either the D arm or the T stem of the tRNA that can sort of, force the tRNA to stay longer time into the ribosome so that this would facilitate the promotion of the peptidyl transfer reaction so that we could possibly see a better elongation, reaction when we're trying to incorporate these gamma amino acids. So just to sort of optimize the reaction condition, we optimize a range of elongation factors from elongation factor G, EFP, and the EFTUTS. So all these elongation factors have been optimized so that we could produce the highest concentration of the translated peptides. But we also optimize the concentration of AATRNA so that it could also provide us with the best optimal translation reactions. So one interesting fact that we observe is that when we are using the best of the best condition from each optimization, we actually seen a additivities that's being observed. Basically, all these factors, we didn't see sort of like negative cooperativities, but each factors they sort of like add up to the translation efficiency one by one. So I think that sort of like address some of the mechanistic side about how these elongation factors are actually helping with ribosome translation. So with that optimal condition, we then sort of move on trying to test on different codon like compatibilities. So because we don't want to just use one set of codons, right? We wanted to provide a set of codons that people can use. So with this phenylalanine-like gamma amino acid, which we call them frucro. So the F stands for phenylalanine, and the cro stands for crotonic acid, which is basically this gamma amino acid backbone. So we use that to test out three different set of codons, and then within these codons, we see like pretty good translations of the gamma amino acid. We also chose this leuco. Basically, the L stands for leucine, and then cro stands for crotonic acid. So with leuco, we also see pretty good translation happening even after we switch to different codons. So I think with this set of experiment, we're trying to see if we can use two different codons to incorporate different gamma amino acids in one peptide elongation reactions. So, and then we did test that, but here we didn't test different codons, but with one codons, we trying to separate them by different lengths of amino acid just to see what's the, like the maximum comparabilities of these gamma amino acid. Okay, I see the chat box. Sounds good. What we see is that these side-by-side incorporation is not possible. Alternatives are also not possible. But once they are separated by two amino acid residues, this multi incorporation become possible. So that we know the pattern that this gamma amino acid has to be separated in a minimum of two alpha amino acid residues. Otherwise, we won't be able to see any products. It's just all truncated product. So this is just to test the limit. But we also tested other side chains, like other gamma substituted amino acids. We have Vicro, which is the valine look-like protonic acid. Also the Ascro, the serine protonic acid. Also the Acro, which is the alanine substituted, the single methyl substituted protonic acid. We also just have the very flexible like which I call it G-Crow because it looks like glycine. So this basically is a freely rotated, well compared to other gamma substituted protonic acid. This G-Crow is like the most flexible residues among all the residues that I tested. And then I also observed some BME and DDT adduct, which sort of is a sign that this residue itself is kind of more reactive towards the cysteines or any free thiol that present in the translation system, because we always have some, content of BME and DDT in the translation mixtures. I also tested a like R-Ascro. The little R stands for the D amino acids. Basically, this serine here is the chiral inverted version of the L-serine. Honestly, this like these chiral inverted residues are kind of difficult to translate it, but with some efforts, I still got these chiral inverted protonic acid being translated. So that's very excited to see this could happen. And then overall speaking, I also do a semi-quantitative quantitation on the translation node. So before and after the optimization of translation systems, adding the elongation factors, roughly see a twofold increase in terms of the translation node with these range of unnatural amino acid. So I think adding these elongation factors do actually helps with the translation load. So and then it is a must-to-have component when you're trying to translate unnatural amino acids. Um, so some of the application I'm trying to sort of like show by using the crotonic acid is to, like the key question is whether we can make it to macrocyclic peptide so that it will be compatible with the rapid discovery, which I think Peptide Dream and Suga Lab we have been used for the last 2 decades to generate microcyclic peptides. And as since the microcyclic peptide become more of like interest recently, so I think this would be very important to see whether we can sort of generate these microcyclic peptides, right? So I just put the phenylalanine like protonic acid in the middle of the peptide, and then in the initiator, I use the chloroacetyl D-tyrosine. And then at the very C terminus, I put, well, it's not C terminus, but close to C terminus, I have a cysteine, and then I'm just trying to see if I can see the cyclized product. So, and then it actually, we observe like pretty nicely the microcycles like mass spec, and it actually corresponds to the correct cyclized product, not the non-cyclized product. And also trying to incorporate F-pro in a more complex, modalities, for example, protein. So here I substituted tyrosine codon with the F-pro. And then you can, after incorporating the F-pro, you can see the mass shift between the wild type of the protein versus the F-pro mutated protein. And then nicely, this is a very nice, like delta equals to almost 10 shift. So lastly, since I mentioned early that we observed the DTT and BME adduct of the G-Cro, which is the flat, more slightly flexible crotonic acid. So I was just wondering if I chose G-Cro as the initiator and I put a cysteine and also in the middle of the peptide, whether I would see a self-cyclized product since this residue itself can react pretty good with DTT and BME without any like any additional component or additional nucleophile adding, and then it's at pH 7.5. So I just run this reaction, but then I put the G-Cro as an initiator, and then I do see observe the desired product, and then the desired product can no longer be modified without acetamide, so I can confirm that this, cyclized product has actually been formed. So with that, lastly, I just want to touch a point. So some of you might wonder whether we use this residues in the random non-standard peptide integrated discovery, which is the rapid system. So actually we did, and then the draft is in preparation. What I can, prelude a little bit is that these very interesting, these crotonic acid contain peptide display picomolar potency and well in bindings and also in cells. And then these actually can be translated into microcycles, and then without them, the binding affinity will drop at least by 200-fold, and then it will no longer display potency in cell assays. So yeah, lastly, just I need to thank greatly towards Professor Hiraki Suga, like because in here like he gave us full support on anything that we wanted to do, and then it is such a friendly and very helpful research group. Like I truly learned a lot and I appreciate a lot. So yeah, that would be everything from my side. And then I can take any questions if you have any.
Great. Xiyun, thank you so much for a fantastic presentation. We have one question so far, which was a question that I have, but I guess it was inspired by potentially your sort of not a covalent reactive work that you did prior to this work. And that was, sort of the concern about even though they're in natural products, the reactivity of these things and potentially
Mhm.
needing to engineer them out. And so you've also shown that you can use that as an advantage in a way to do the cyclization. And so I guess it answers the question of, are they reactive? But I guess we need more information on that. I guess the question I had related to that while we wait for additional questions is the cyclization stereospecific? Because you're generating a stereocenter, and have you been able to determine that at all?
No, so I wasn't able to determine. So I don't think this is stereoselective, but I so with the one of the building blocks we actually did, like we use some other nucleophiles to do addition, like a range of thiol nucleophiles to run the addition, and then we do see a roughly 2 to 1 or 3 to 1 ratio in terms of the thiol adducts on the LCMSs. So I think it really depends on well, one is the chirality of the thiol, and then the um, well, like the structure of the thiol reacting partners. So I do think it's possible that we have some stereoselective, but here since I didn't add any, chiral catalysis, so I didn't control the formation of the adducts. So here for well, just for these cyclization specifically, this is just, like based on the observation. So I run this experiment, but I didn't expect it to be chiral selective.
Okay. Yeah, that's
Yeah.
that's fantastic. So it should be a potentially a development challenge for the med chemists when they get their hands on the hit to lead and lead optimization of these incredible looking molecules. There's a second question on the concerns of immunogenicity, if you could answer that as well.
Mhm. Immunogenicity.
These
Um,
of these peptides, correct? Yeah.
Interesting. So, yeah, I think that's a great question. So I actually haven't thought about immunogenicity because we haven't yet gone to, to that part with our like crotonic acid bearing peptide inhibitors. Um, I think my major concern is that it might actually got uh, sort of like reduced by free thiols in the bloodstream, but whether it would generate immunogenicity, I think it would largely case by case, since the crotonic acid that we provided actually. So here I showed roughly 7 kinds of crotonic acid when you're having different gamma substitutions. So I envision that different substituted crotonic acid may have different immunogenicities. So I think that will still largely depends on this also the full sequences of the peptide. So just this one building blocks may not be, like big enough to impact the immunogenicities of the peptides, I think.
Right. Okay. Yeah, and I think, obviously these molecules will need a lot of optimization, likely that is, in a sort of a lead op, and those issues will be answered. Then a lot of people will perform those immunogenicity experiments in order to determine the liabilities potentially of these peptides. Just one quick last question while we have time, and that is that you work both in the Pentalutin lab on ASMS and then now on the rapid system in the Suga lab. Could you compare the two in terms of do you seeing false positives or false negatives with either of them?
Mm. So the key differences between the MR display and ASMS is that for ASMS we run one selections because peptides cannot be like they're not DNA, they're not RNA, they cannot be amplified. So for ASMS we can only run one selections. But for ASMS we use a very stringent way to separate the non-binding versus the binding peptide from the protein. So and then with ASMS, I think because of the selection is so stringent, so that I think most of the time we see like it's just positive. Like false positive is I don't see a lot of that during my time or at least on my projects. But MR display is sort of like you have a mini evolution goes on in a lab. So basically, for MR display, you're running like so a few cycles, and then for each cycles, you're using DNA barcode, and then you re-amplified these high affinity binders. So during that, cycle of amplification, I think these the sort of like involving process would sort of wash out a lot of false positive signals. But a lot of times I would the false positives like signals or false negatives, well, these signals are actually bead binders. So you can tell by the negative selections because we usually we do sequencing on both positive and also negative selections.
Right.
So by comparing these two set of data, you can, fish out some of the noises
Right.
in your poster. So
Yeah, this it's a great question that probably we need a lot more time to answer and discuss. So in the interest of time, I guess we'll move on. I'll let Danielle introduce our next speaker. Thank you so much for your
thank
presentation.
you.
Yeah,
My
thank you,
pleasure.
Xiyun, for a great presentation. I'd like to now move on to our second speaker, Dr. Genwei Zhang. He's the head of peptide platform at XtalPi. And as he grabs his slides, he'll be speaking on AI-enabled next-generation cyclic peptide drug discovery.
All right. Thank you, Daniel, for your kind introduction. And can you guys hear me okay?
Yes, I
Just
do.
making
Yep.
sure. Okay, cool. And good morning for folks dialing from the US, and evening for people. I saw some of the folks attendees from China and, or Asia area. My name is Genwei. I'm the head of the peptide platform at XtalPi. So today, I'm very glad to have this opportunity to talk to you a little bit about, PepX, which is our integrated peptide discovery platform. This is, driven by AI intelligence, automation, and high-throughput library screens. So we also, accumulate a lot of experiences for cyclic peptide discoveries. So today I will also delve into this direction and share you a little bit of, about the case studies that we have accumulated on this platform as well. Before really talking into the platform technology and case studies, I want to just go over a few high level slides to talk about this field and the development trend. So we know the peptide market is surging. The peptide blockbusters and emerging oral peptide molecules really changing the commercial landscape and opening new opportunities for new modalities. So the GLP-1 drugs apparently has transformed the obesity care, but I think this might be just the beginning. So we have seen also more than 10 other oral obesity drugs being tested in late stage clinical phase 2 or 3 trials. And furthermore, people to avoid crowdedness and to make the drugs more efficacious, they try to involve more targets. For instance, amylin receptors. There's at least more than 15. I have seen more than 15 pipelines involving amylin. These pipelines being tested in late stage clinical trials as well. Amylin receptors actually been validated by Eli Lilly as well. So we have seen this field on the left hand side, GLP-1 or obesity care field has been very crowded. As you can see, these blockbusters, tirzepatide, semaglutide, already exceeded the antibodies, Pembro or others ranked the top and second. Especially very interesting, the tirzepatide, this dual target molecule has exceeded semaglutide last year in terms of the global sale. Potentially could be the fastest ever growing drug in the history, which is a huge. But when people saying, okay, if we want to still enter or further expand in this field, do we still have opportunities in the GLP-1 field? That might be, I think, still some opportunities, but might be very challenging unless you can address really the problems there. For instance, the lean mass loss, the side effects, the patient compliance issues. But I think will be very challenging. And then a lot of people also when they talk to me and see what would be the new opportunities for us, I think what is driving the next wave will be the cyclic peptides. Right? We have seen earlier. On March this year, we have seen FDA approve Johnson & Johnson's oral peptide molecule 2113 for treating psoriasis, medium to severe psoriasis. I think this although we have more than 80 peptide drugs approved by FDA so far, but this one is a huge milestone for people, working with cyclic peptides. This is, orally bioavailable, although it's only 1 to 2% the bioavailability, but one single molecule can reach picomolar binding affinity and it can compete with antibodies. I think that's really amazing. And this is not a single case. And this Merck's molecule also completed its pivotal clinical trial and overcome its oral delivery barrier and positioning itself as a potential blockbuster as well. We have seen these successful cases, I think, thanks to the technology advances, not just serendipity anymore. So apparently we have opportunities there. We said this peptide therapeutic market is really, growing very fast. Here I borrow a forecast from Grand View Research. They made this prediction that this total peptide therapeutic market is projected to reach over almost 300 billion by 2033. I think this is enormous with a growth compound annual growth rate almost at 10%. The Novartis executives has publicly made this statement that macrocyclic peptides in the next 5 to 10 years could be a top drug modality to lead this oral biologic era. I think the MNC companies now really accelerating their pipeline expansions on peptides. One is through internal innovation, and externally will be collaboration and partnership with this AI peptide platforms. For instance, here give you a few representative example. The AstraZeneca collaborated with a China company JSPS to drive innovative peptide molecule development. This upfront is huge. This probably a hybrid collaboration, not only platform partnership, but also including licensing. Novartis collaborated again with the UNP's AI platform. AstraZeneca also collaborated with a Chinese company, Syneron, last year, I think, to discover oral cyclic peptides. I think this is just a few examples to represent. So I think when we talk about cyclic peptides, the core value of cyclic peptides, I think it's a matter of molecular game. It's a matter of molecular game. So small molecules has been out there for over 100 years, right? And it's perfect to penetrate, diffuse, no bioavailability issue, but it only covers a small portion of the protein targets, 15%. It requires well-defined binding sites, and then oftentimes has off-target side effects. For larger biologics like antibodies, don't have such an issue. Usually high, turn out to be very high binding and it's very selective, but it cannot penetrate very well. Although it covers slightly bigger portion of the protein target, but overall 30%, mainly ultracellularly. So I think together these two only 45%. So the intracellular targets lacking well-defined binding pockets, 55% conventionally regarded as undruggable. And cyclic peptides now lies in between small molecule and large biologics, can potentially address this portion of undruggable targets as a novel solution. I think inherits a lot of advantages from both molecules, high selectivity, big motor binding affinity, and potentially penetratable. I think this is making it very attractive. And last but not least, all these targets that are being validated by small molecule or antibodies potentially can also be, again, be developed with peptides as well. Like we have just talked about the Johnson & Johnson's molecule, right? It can now start eating up the antibody dominating the field, autoimmune diseases. Platform. Essentially, our vision is this is an end-to-end platform. We want to turn our AI insights and years of peptide therapeutic experiences into more potent, stable, and clinically viable peptides, hopefully faster than ever before. Our goal is to compress this preclinical candidates discovery duration as short as we can, hopefully within a year and a half, sometimes within a year period of time. A typical scenario that we start from a targets.
Genwei, we lost your audio a little bit. Sorry about that, everyone.
Genwei, if you can hear us, maybe
Yeah.
take your video off in case it's a bandwidth problem and see if that helps bring back the audio. Yeah, still we have
Yeah.
difficulty. We don't hear you at all, but we can see your mouse moving.
What about now?
You're back. Yeah.
Okay. I'm sorry, my internet got interrupted.
We don't
Let me
see your
reshare.
screen now, though. Oh,
Yeah,
there we
he's going to share again. Yeah.
Yep,
Yeah,
perfect.
I paused. I paused the sharing.
Yeah,
Okay.
perfect. We can see it now.
Did I get a? So I was
You
got
started
cut
with
off on this
targets.
page.
Yeah, if you go back to you start with targets,
Okay.
that's where we lost you.
Okay.
Yep.
Okay. Cool.
Thank you.
Oh, sorry. Oops. No. Yeah. Thanks for the reminder that I didn't realize the internet was cut. So yeah. So as I said, so we can start with the target of interest and rapidly perform hit discovery and lead optimizations and all the way to recommend preclinical candidates. So this iterative cycle from dry and wet, making this working very smoothly, and across the board on the right hand side, you can see we have validated in different indications and diseases this platform capability, like first in class, undruggable targets to treat renal carcinoma, weight loss peptide, radiopharmaceuticals, brain ocular delivery, oral peptides, and etc. So a lot of people talk about iterative dry and wet cycles, but how do you make this dry and wet part working seamlessly is really the critical part. So we have done this many years and accumulated a lot of experiences on doing this. And importantly, another differentiator on this platform will be a non-canonical proprietary monomer that we have been built. So this is not a simple derivative of the naturals or the one that you can find directly on the market. So we have spent a lot of time to curate it. There's a lot of information we could borrow from small molecule space. The diversity of these non-canonicals currently available is certainly not enough to cover enough chemical space. So we did a lot of work on that as well. So now XtalPi have built a proprietary more than 2,000 non-canonical amino acids to expand this druggable space searching, and this is very efficient. From the source, we can solve a lot of challenges to find a high quality peptide instead of leaving this a lot of work to later stages during optimization. From another technical perspective, as you can see, we synergize AI and high-throughput library screen optimization to accelerate this early stage discovery. At the AI part, we have large pre-trained peptide models, structure prediction models, and also generative predictive models to support. But really, the foundation of these models is the high quality larger mod protein peptide interaction data. And also the proprietary non-canonical amino acid library help us to break through the bottleneck of design. When we after designing up, when we turn into the wet lab for verification and optimization, definitely automated synthesis can bridge these two. So parallel solid peptide synthesis we typically utilize, and then if there is a need for further optimization to improve its drug likeness, so we have also built this messenger display library, but certainly not that capable of incorporating non-canonicals that what Xiyun or Suga's lab has done over the years. But to really complement that weaknesses, so we also have this, chemical combinatorial library approach, OBOC. So this can definitely unlimited incorporate any, non-canonicals at any position as you want. Although the library diversity is slightly, lower, but I think these two together can seamlessly work together to fine-tune and optimize the, your peptide molecules. And lastly, for scenarios that you don't have a specific targets or your targets is undruggable intracellularly, we can also perform high throughput phenotypic screenings, directly screen large peptide library on a functional cell assays. This is also a matured workflow on this platform. So next, I will quickly give you a few case studies that this platform have already done and shown. So the first case is a de novo design on this platform. The disease we chose was a neovascular AMD, which likes very effective therapies. This protein we choose has been well validated. It's implicated in this disease, and what we did is on this identified binding epitope, we know when the molecule binds here, it can block the function of this protein. We rapidly discovered this hit molecule through our pipeline. As you can see, our AI model can generate backbones, typically tens of backbones to sometimes maybe several hundreds of them. On each backbones, our models can fill the sections and evolutionarily mutate them and then make the best predictions. And after generative AI, the predictive AI will come up to evaluate its binding affinity, stability, immunogenicity, solubility, and etc. There's many features as different models support these different predictions. But at the early phase, you probably care more about binding affinity and stability. In these cases, to simultaneously improve binding and stability, we also incorporate non-canonicals since our model prediction is a full atom, so we can basically incorporate any non-canonicals. When we resynthesize and test these design sequences, we can see a hit generation success rate over 10%, which we think, in comparison with the conventional, designing approach, which is very significant. And this 22 nanomolar binder macrocyclic can be was discovered in this case. The first case is a de novo design case, and oftentimes when you find a hidden molecule, you still need to optimize it further to improve its its pharmacological features. So in this case, that we work with our collaborator to optimize their hidden molecule. This protein is a dermatitis protein target. What we choose is our AI modeling and runs of OBOC screening. We simultaneously increase its binding affinity as well as stability. I didn't show the workflow is quite straightforward. You build this library. Actually, it's a focused library, but where and which molecules you should mutate. This needs some information or rationale from your modeling. But here you need to weigh, okay, which, if your model is more convincing or giving you more confidence, you probably can, rely on more your modeling output. But sometimes if, your modeling, your complex prediction isn't really stable, you probably can leverage more from the medicinal chemist experiences. But together, these experiences can build into your focus library, and then after screening, further enrichment, you can improve your binding affinity or stability. I didn't show we also found in this case single-digit nanomolar binders, but we didn't show it because of confidentiality reason. But overall, we have seen that one round of library optimization, we can improve the binding affinity over tenfold, and also the plasma stability 5 to 10-fold as well, and that has been consistently proven on this platform. So I talked to you a 0 to 1 de novo design case and also a optimization case. Real projects here, I'm showing you that we can consistently produce and output this binders, cyclic binders. So this is a targeted brain delivery of antibody. So tissue organ specific delivery apparently is very needed in the therapeutic application, especially in some challenging areas like a brain disease treatment. So talking about this brain disease treatment, here we choose the indication Alzheimer's disease, which is very challenging, difficult. There's only 2 approved antibodies in the field, Eisai Biogen's lecanemab and Lilly's donanemab, but both of them have very severe concerns of safety issues. So especially the area E and area H causing brain swelling and bleeding. The area can be very severe or fatal. Actually, in a clinical trial, this antibodies cause patient death, and that's why although lecanemab, donanemab got approved by FDA, but not by other agencies like EMA. Still have concerns on this antibody, and they use very high dosages to treat the patients. I can't remember exactly the dosage, but the reason why you have to infuse or inject the patients such a high dose of antibodies is because of the barrier, the BBB barrier is there. So although later stages now Roche and Denali has validated, for instance, leveraging transferrin receptor, we can improve the benefits of the patients, but still the area side effects is still there. So our vision is here to develop a peptide warhead that can efficiently deliver these large macromolecules into the brain, and hopefully that we can increase its brain exposure and reduce its dosages by, for instance, 5 to 10-fold, and that way we can bring up very effective and safer AD treatment. So we still leveraging receptor-mediated mechanism. Here it could be single or dual targets. Transferrin receptor, people have spent a lot of research and validations on transferrin receptor, but using antibodies, but not for peptides. We still need to spend a lot of time to optimize its binding affinity, its penetration. When we validated in vivo, we've seen that our molecules can expose very obviously in the brain, but this dynamically it's very fast. So now we are leveraging also dual targets to hopefully have the least perturbation on the PK profile, but it gives the maximal exposure on the brain. So I think this is in vivo validation still in progress. These binders have both linear peptides or disulfide bridged peptides for better CMC, as well as some other cyclized peptides, which needs chemistry to conjugate these molecules onto antibodies. But we also at this stage conjugating with the nucleic acid like siRNA molecules to further demonstrate its delivery effects. So the case 4 is the radiopharmaceutical.
Sorry to interrupt, Jen.
Yes.
We have about 3 minutes left of the webinar. Yeah.
Okay. Yeah, I'll go quickly, close up in 2 minutes. So radiopharmaceutical is, in this case, we choose a tumor, very highly expressed tumor associated antigen, and we discovered this is collaborative projects. Our collaborator provided this target to us. On our Genwei experiments, we discover rapid picomolar binding cyclic peptides. As you can see, 100 picomolar binding affinity. But at this binder, we still need to improve its stability through non-canonical amino acid incorporations. So early stages, although the molecule after cyclization optimizations, it can have a isolated plasma half-life almost 4 hours, but we might need a even larger half-life time. Here after our optimization, now we have reached over, actually this is not the best molecule I'm showing today, hasn't been updated, over 50 hours almost. So I think this is non-canonical amino acid incorporation can help this. So case 5 is undruggable targets, IDP, very challenging, conventionally regarded as undruggable transcription factor. But on this, XtalPi developed a unique wet, Genwei combo approach to tackle this problem. So I think we also discovered a 100 nanomolar cyclic peptide binder, and it shows very good cellular efficacy. Now we are, optimizing further its penetration to further improve its therapeutic window. I think this is, really exciting to see. Okay, undruggable targets, we can also discover peptide binders. For oral peptides, I probably don't have enough time, but our strategy overall is to not only improve this molecule stability at the molecular level, but also using formulations. We have seen we can, improve at least 2 folds, for instance, its bioavailability in comparison with Novo Nordisk, semaglutide. I'll skip this. This is our pipelines. The 3 directions that we are hopefully can drive and reshape its innovation is drug delivery using peptide as a warhead, also oral peptides and the cyclic peptides on this platform, which is driven by intelligence and automation. So last but not least, XtalPi is an AI and robotics technology platform. We use our AI and robotics as a infrastructure and enabling technology to solve challenges in not only life science but also in other areas like material chemistry or agriculture, energy, and etc. This company founded in 2014 by 3 postdocs at MIT. From a very niche area, crystal form prediction, we further expanding our businesses to small molecule, large biologics like antibodies and peptides and nucleic acid. And now, we 2024, we went to public, Hong Kong Stock Exchange. XtalPi is a global company that we have our R&D and mainly done in mainland China, Shanghai, Shenzhen, and Beijing. I'm based in Beijing. And also we have a global business center at Boston. Liverpool, we acquired a company last year and now expanding our footprint in Europe as well. We work with the leading life science companies, MNC companies, and the big biotechs worldwide. Overall, we have 400 customers globally. This is at least a few MNC companies and also some representative biotechs. So this is the last slide. So we welcome you to collaborate with us via various cooperation channels, either some asset licensing or a co-development mode through joint discovering the program. So with that, I wanna thank you for your attention. I'm sorry for this taking a little bit longer than expected.
That's okay. We had some
questions?
technical difficulties. Yes. Yeah. So there's quite a bit of
There's
questions.
Interruptions in the middle.
Yeah. If everyone's okay to stay on the call, I'm happy to extend the webinar to get through some of these questions.
Yeah, I think we should. And Genwei, thank you for a wonderful presentation. There's some impressive case studies there that I'm sure people have many questions on. So yeah, happy to stay on and answer some of them. Just a quick comment in the Q&A. you can vote, I think, with the thumbs up, and that'll shift
Yep.
the answers in terms of a priority. In the absence of that priority, I currently will take them in the order that they were received. And I think some of the challenges with both AI and kind of reproducing things from de novo screens and or computational approaches is sort of the synthesis challenges. And maybe I think one of the questions is how do you interpret that, and then what's the success rate and sort of the yields associated with the early kind of hit to lead compounds?
Like, is the synthesis yield or the
Yeah,
hit
so
generation?
if you could
Sorry.
comment on the success rate and synthesis yields
Oh,
from those
synthesis yield.
efforts. Yeah.
Yeah, okay, synthesis yield. Well, I think for it depends, how many non-canonicals or how many, for instance, methylated monomers you incorporate in your molecule. Sometimes could be very challenging. We have to first, pattern test to process and see how, what's the best condition, and then come back to run again. But for simple, if there's no methylated ones, not a very large set chains, and then the typical yield would be quite high, actually 20-30%. That's very, very common. But for low yield, low synthesis yield molecules, you have to really, you know, optimize the condition. Yes.
Yeah, it's a general problem actually, and it would be
Yeah.
nice to see if the AI and these perspective designs can actually predict synthesis success as well. But that's a maybe a topic for another webinar. I
Yeah,
guess the
we always
other
got these questions, but yeah,
yeah,
sure.
the other webinar, I'm sorry, the other questions related to this actually is sort of the hit rate compared to other AI approaches such as Bolt Gen. There's a suggestion that there's a case study that the 13% success rate, and if you
Yeah.
could comment
Yeah.
on yours as well.
Yeah, I don't know, to be honest, the Boston's success rate, the 10% or even more or less. But this is just one case. We had another, I didn't get a chance to explain. We had another tumor vaccine program, which we trained this model with over 17 million of peptide MHC1 molecules and 4 million peptide MHC2 molecules. That predictions success rate is over 50%. So the really the value in that case study is the data. So in general, peptide protein interaction predictions, I think 10% now we are really at a high standard already, I think. But if you have high quality, even larger amount of protein interaction data to further train and fine tune your model, I think there's still room we can further improve, because in our AI model validation, we have seen that there's still a big room to improve in terms of sequence recall accuracies. But I didn't get a chance to delve into too much technical details. But I think yes, I think I don't know. We definitely look forward to talk more with this audience
Yeah.
and see how Boston's success rate would be. But I think yeah, it's a challenging problem in the field. Yes.
Yeah, I guess related, and I've been able to update my view in terms of the highest voted questions. And so here's the next one, which is really, can you comment on the ability of your platform to provide hits for targets that do not have X-ray crystal structures available?
Yeah, so that intrinsically disordered protein project was one of the representative one that we don't have too much computational, work that can be referenced because, it's fluffy. Even if we use AF203 to predict, there's a lot of fluffy and loop regions which you just can design, your binders use a computational approach, right? So we have to leverage that limited information and with our library, and then after 2 runs that we found good hits. So I think the key thing here is that how efficiently you can leverage Joanna West circle. There's multiple chapters in the loop, right? Even one single loop can affect your whole circle's efficiency. How fast you design, how, accurate your model predictions are, how fast your data can be generated and feedback, all these matters. In that case, that we, 2 rounds of, design and data feedback that give us a lot of confidence. Eventually, we did design that 100 nanomolar, cyclic binder. I think, yeah.
Yeah, and I guess related, the next question was about sort of how impressive it was to find binders to the intrinsically disordered proteins. And was there, you kind of went over it, but was there anything special in terms of how that was achieved that you did?
Yeah, so you should have a like a foundation model or a model that already learned all the information, sequential information, structural information, your protein peptide interactions. And then when it comes to a, intrinsically disordered protein, and then there might be some of the residues that have better uh, uh, possibility to bind on certain regions, but you have to through a large library screens on the wet lab and then find them and then feed the model fine tuning to let them, okay, have the test. Okay, now we need to predict more binders toward that direction, then maybe it can help. So this is, our feeling, but it might be still a black box, right? How it works better towards that case. Yeah.
Yeah, that's excellent. And I mean, this is maybe a similar question on, the challenges of synthesis and just other properties that are predictive. You mentioned solubility once at one point, or maybe not solubility,
Yeah.
but stability. And maybe you could comment on prediction of solubility or things like aggregation or even on the chemical side, polymerization in some of the synthesis due to the non-canonicals.
Yeah, that's a great question. So non-canonicals, it's challenging to predict their aggregations and solubilities. Right now, we have a lot of information that we learned mainly on the natural space. Yes, on natural space solubility predictions, we have seen very high, and the predictors we rank them with highly soluble or medium soluble, moderate, and hard, insoluble. So as long as they can predict into the right category, that we think they are, good. So it's not very precise, how the solubility values is. So I think that it's challenging, especially for non-canonicals.
Great. And I think we're getting close to, I think, sort of the
A lot of questions.
questions, but there's a lot of themes here, I guess. And so
Oh,
maybe what I'll
wow.
do is, I guess part of it is really this sort of comparison, perhaps, between a computational prediction and the experimental reality.
Yeah.
And maybe you could shed some light on that a bit.
Um,
I
Sorry, I
know
was looking through I just opened the Q&A window and
yeah.
went through so many questions.
So
Yeah.
yeah, I
Okay.
mean, there's some related questions around, sort of how is AI used in the design workflow? Sort of like between, what is the how does the what's the reality between that in the experimental reality and what you do that relates to the questions we had about success rates, for example.
Yeah.
So, I think these are topics that we could talk a lot more about. Maybe they're not very specific questions that can be answered quickly here. So maybe what we'll do in the interest of time, and given that we have extended the Q&A, is wrap up the Q&A for now. I'm sure we can connect you to the folks in this chat. Maybe Danielle, you can comment on that.
Yep.
Genwei, yeah, go ahead.
Through the registration, I should have, and that I see the questions. So they'll be captured and I can send direct email links, to introduce and we can try to get these questions answered.
Fantastic. Okay.
And then,
Sure. Yeah.
the other thing to note as well, this webinar has been recorded, so it will be available on the PDHC website. After I should hopefully have it up by next week. And I think through your registration, you submitted your email and name and company information. So XtalPi would be able to contact you if you wanted to collaborate further or ask any additional questions.
Fantastic.
And I just want to thank again XtalPi for doing this webinar for the Peptide Drug Hunting Consortium as one of our legacy sponsors. Our network is a nonprofit organization, 501c3 organization, where we look for direct donations to be able to host these webinars and our other events. And we have a lot of fun initiatives coming through. All right. Thank you, everyone, for joining.
Thank you.
Thank you to our speakers, and we look forward to seeing you at the next webinar.
Excellent. Thanks, everyone.
Thank you.
Thank you very much.
Thank you.
Bye. Thank you, everyone.